A very warm greeting to all of you. It is my great pleasure to share with you our talk on an overview of molecular marker for identification, taxonomy, and phylogenetic study of eucheumatoid. This talk in general can be divided into three main parts. Firstly, on strain selection for mariculture, followed with molecular markers for taxonomy, ID, phylogenetic study, and strain selection. And I will conclude the talk by giving the suggestion and on whether is molecular identification all we need. Okay, all of us are aware that seaweeds are economically very important. It worth more than 6 billion USD per annum, and seaweed, whether through farming or forage, can be used in many areas such as food, being extracted for phycocolloid, herbs, feed and fertilizer, and can be used as bioremediation, and potentially can be used for carbon sequestration. So for Malaysia, eucheumatoids are very important because this is the only two species that are cultivated commercially in Malaysia. These seaweed are regarded as high-value commodity in the 10 and 11 Malaysia Plan and contribute approximately 45% of the country's total aquaculture production by weight in 2019. However, we face challenging is with regard about production, where we have saw the decline of production, in which in year 2012 there's a total of 331k ton being produced, but in year 2019 only 188k tons was being produced. One of the factors for the decline is probably due to the aging cultivar of KAPPAPHYCUS erasea, which have been vegetatively propagated for over 50 years. Okay, when we talk about aquaculture, strain selection is always very important. Selecting a good cultivar or strain will improve crop performance and yield. And strains are often selected based on economically important traits such as fast growth rate, high survival rate. In this case, it is very important to be disease resistant, high yield of desired product. For example, for eucheumatoid is the biomass of carrageenan, and also with the increased phenomenon of climate change, the crop must be resilient toward climate change. Strain selection is conventionally done by selecting promising individual from the wall, followed by calculating, crossing, or hybridization to produce quality offspring. The procedure usually spanning multiple generations. However, the strain selection of eucheumatoid is rather challenging. Firstly, it's due to the seaweed are morphological plastic. Individuals of the same species may look very different from one another and vice versa, which they are existing of different morphotypes in a single species. Clonal variation is also common among cultivar, and at the same time, the specimen collected from wall can be damaged or deformed and difficult to morphological assess in terms of the qualitative trait as well as for identification. Selection may be facilitated using DNA technology, in which selection can be done more reliably based on intrinsic property of seaweed instead of extrinsic one like morphology. This is done by identifying or analyzing specific DNA region of the seaweed known as DNA or molecular marker. Okay. Strain or species selection using molecular marker is often referred as marker-assisted selection or MAS. Strain selection and taxonomy are often linked to each other. Okay. Molecular identification and taxonomy. DNA data can be used to differentiate seaweed that are highly phenotypic and ecotypic. It can also be derived from telefragment or young or damaged seaweed. In the case of eucheumatoid, the correct identification of seaweed will allow farmers to predict the nature and property of a component. For example, carrageenan type, in which KAPPAPHYCUS produce kappa carrageenan, while eucheuma produce iota carrageenan. Okay. At the same time, if we can also identify the quantity trait locus or loci or QTL, okay, finding genetic region or loci that link directly to economic importance traits like high growth, high carrageenan yield, and high gel strength, etc., can reduce the time needed for strain selection. This can be done also by using molecular marker. Okay. What are some of the genomic tools for identification and selection? Strain selection and taxonomy are often linked to each other and may utilize the same technique or technology. Some of the conventional techniques for strain identification and selection are random amplified polymorphic DNA technique, which is in short called RAPD, restriction fragment length polymorphism RFLP, and amplified fragment length polymorphism or AFLP. Newer techniques involve linking digital DNA information to economic traits such as DNA sequencing, microsatellite, single nucleotide polymorphism or SNP, or marker-assisted selection, MAS. This is the table showing the overview of genetic approaches used in molecular taxonomy for SEAWEED. Okay. From late 1960 to 1970, generally the main approach using are protein electrophoresis applied to isozyme and allozyme. In late 1970 to 1980s, RFLP analysis of mitochondrial DNA. Mid-late 1980s is DNA fingerprinting, for example, RAPD. 1990s up to present with DNA sequencing, AFLP, single-strand conformation polymorphism. As 1997 to present, there is use of genome approach such as express sequence tags, DNA sequencing, multigene, and phylogenomics. SEAWEED taxonomy. Identification and species delineation is problematic due to lack of distinguished morphological characteristics and plasticity of SEAWEED. Breakthrough in biochemistry and molecular biology have caused a new development for algae systematic. Okay. In general, psychologists started employing molecular approach for SEAWEED taxonomy in the late 80s. Some of the earliest papers, example are like Olson et al. 1987 on the green SEAWEED, Lim et al. 1983, 96, and Fenn et al. 1998 on the brown SEAWEED, as well as Goff and Coleman 1988 on red SEAWEED. Taxonomy study is important to ensure that correct species are identified and recognized in order to fully utilize commercially SEAWEED resources and enhance the development of both polyculture and the SEAWEED product processing industry. For example, strain selection to find a fast-growing good-grade algae producer is very important before embarking on mariculture, because variable quality of algae obtained from Gasserilia may present a problem. Now let us move into EUCHEUMA and KAPPAPHYCUS. We know that EUCHEUMA and KAPPAPHYCUS, despite that there are only very limited species available in the world, but they do exist into many different morphotypes, and they are highly plasticity, and the identification is very challenging. Okay. KAPPAPHYCUS and EUCHEUMA are known to be morphological plastic and difficult to identify based on morphological observation only. As we can see that in this picture that they exist in different color as well as morphologically. Okay. Let us look into the overview or historical account as regard about molecular taxonomy of EUCHEUMA KAPPAPHYCUS. A comprehensive study on KAPPAPHYCUS and EUCHEUMA was pioneered by Zuccarello and co-workers in year 2006, where they have characterized DNA of the SEAWEED using COX-2 trace spacer and rubical spacer molecular marker. The work by Zuccarello and co-worker has inspired Tan and co-worker from Malaysia to employ more molecular marker, which is COX-1, COX-2, COX-2 trace spacer, rubical spacer, and RBCL for the identification of eucheumatoid SEAWEED from Malaysia. Molecular markers differ in terms of the quality and to be assessed and decided which to be used internationally. Identify the correct species of eucheumatoid will allow the farming of the monospecies, which is more efficient and producing only a single carrageenan type. This is important as different carrageenan types are processed differently. Based on our study Tan et al. 2012 and 2013, it shows that despite of the various local variety of KAPPAPHYCUS and EUCHEUMA being collected with different color and morphotype, actually majority of them only fall into the following species, such as KAPPAPHYCUS ourasia, KAPPAPHYCUS striatus, KAPPAPHYCUS malaysianus, which is always being thought to be KAPPAPHYCUS ourasia, have been found to be a new species through the molecular identification, and EUCHEUMA denticulatum. As regards about genetic diversity of EUCHEUMA KAPPAPHYCUS in Southeast Asia, with the main species of EUCHEUMATOID being determined, Lim and co-worker established a collaborative effort with major carcinogen producing countries in Southeast Asia to identify haplotype of KAPPAPHYCUS and EUCHEUMA using the COX-2 spacer and COX-1 marker, and this subsequently had become the groundwork for strain selection until today. The use of molecular marker had led to vast improvement in the documentation of KAPPAPHYCUS in Southeast Asia, either regard about finding new species or new record. For example, KAPPAPHYCUS malaysianus was found as new species in Malaysia, and then in Philippines, more wall specimens being recorded are found in Philippines on KAPPAPHYCUS cottoni and KAPPAPHYCUS enemies, as well as study on the potential of seaweed farming leading to bioinformation. Okay, this is a picture showing the different morphotypes of KAPPAPHYCUS enemies and KAPPAPHYCUS cottoni. Okay, these are wall seaweed that are mainly found but has yet to be assessed in terms of potential as cultivar for Kappa carrageenan. Okay. So now let us look into as regard about how much of molecular technique have been applied in the taxonomy, identification and selection of KAPPAPHYCUS and EUCHEUMA worldwide. We know that KAPPAPHYCUS cultivation have been expanded worldwide, but how much do we really know as regard about the identification using molecular approach? Okay, this is a table showing you the various location or country that have used molecular marker for identification, in which I would say is that majority of the identification and effort come from Southeast Asia, in which in particular in Malaysia, Philippines and Indonesia, as with Vietnam, this through the effort of the study of genetic diversity by Lin et al. And then in Brazil, China, India, Hawaii and Tanzania. Okay, so next let us look into as regard about globally how much do we know and how much work have been conducted. Okay, so those spots that is being highlighted in black color are the known area for KAPPAPHYCUS farming, and based on the table we know there is limited number as regard of using molecular marker in understanding the genetic diversity as well as identification of the species being cultivated. However, with the effort of the Global Seaweed Star program, those have been highlighted in green are the new location area that is covered by the Global Seaweed Star project. Okay, but despite of the effort of Global Seaweed Star, I think we can see that there are still a lot more areas that need to be covered. So we hope that we can have the concerted effort from globally to do molecular typing on all the cultivar as well as also on the wall population. Okay, so what are the challenges and recommendation that you would like to recommend? Despite of the development, the progress on strain selection have room for improvement, especially in increased documentation of wall EUCHEUMA KAPPAPHYCUS population worldwide, as well as development of better molecular marker. Okay, This is because some of the current markers that being used display limited resolution and were not able to resolve the phylogeny of some of the clade of the EUCHEUMA KAPPAPHYCUS. For example, for this clade of EUCHEUMA, there is no support. Okay, so as for other species of the KAPPAPHYCUS and EUCHEUMA. Okay, so at the moment, what are the ongoing research? Okay, there's a need to develop new molecular marker depend on various factors, including genetic variation, able to identify species or better, even up to haplotype, reliability, ease of obtaining the DNA sequencing, affordability, inexpensive method for large-scale sampling, and ability of reference sequences in gene bank for comparison. So this is an example of work by Tan et al. In 2012, where the comparisons of the different marker potentially they can be used for barcoding. Okay. The combined use of COX-1 and COX-3 spacer was shown to display good phylogenetic resolution. Okay. However, the lack of sample species hinder a comprehensive phylogenetic of the study's time. Efforts are now being put into development of testing combination of other molecular marker, as well as encouraging the education and standardized use of the same genetic marker for KAPPAPHYCUS and EUCHEUMA, analyzed by scientists across the globe. This will greatly aid in strain selection. Okay, this is an example of the efforts of through the Global SEAWEED Style Program, in which it involved to multi-marker assessment, in which in this study a total of 9 molecular markers have been tested from the various regions, from plastic, mitochondria, as well as nuclear region. So we hope that this work will be complete soon, so that we can publish the result. Okay, so what are the recommendations in order to help for us to understand the genetic diversity identification as well as phylogenetic study relationship of the eucheumatoid? I think greater effort in using molecular marker on both cultivated and wild species of eucheumatoid should be encouraged, as this will give us better understanding on the species diversity, genetic diversity, and phylogenetic relationship, in hope that through the study we can introduce more haplotype, more species to be cultivated, because currently the genetic diversity or the haplotypes that are being cultivated worldwide, the number is relatively very low, and this is very unhealthy for the aquaculture industry. Okay, and we have seen there's an increased trend in the taxonomy study of other type of species of using phylogenomics, which is organelle genome or multi-gene approach that can potentially reveal more critical species, okay, provide robust framework for inferring evolution, as well as to resolve phylogenetic position of the molecular unusual species, as well as to provide robust phylogeny of the genera. I would like to conclude my talk with the question on, are molecular marker all we need? Can molecular techniques replace traditional taxonomy? Although molecular techniques have been shown to be very useful in the case of eucheumatoid, conventional morphology is still required for quick identification of the field, as well as the determination of new species in which reference DNA is unavailable. Okay, as such, both methods have to be used as a form of polyphasic approach for better accuracy and reliability. Molecular techniques are only accurate if the underlying fundamentals are achieved. For example, correct sample identification with labeling, testing a statistically viable number of samples. Molecular analysis also depends on the marker being used, which in turn depend on several factors such as genetic variation, reliability, affordability, and availability of reference sequences in gene bank. Standardization of the molecular marker being used is important to allow an apple versus apple comparison in research. So with that, I would like to thank you for your attention.